Hi, everybody, and welcome back to a super special session of Session 3, CRISPR Innovations and Technologies. I'm, of course, your panel host, Jared Carlson-Stevermer. I'm the Associate Director of Discovery here at Synthago. And thanks for stopping by for our on-demand videos here for World CRISPR Day. The technology that I'm really excited to talk to you about today is all about spatio-temporal control of CRISPR editing and what we've done at Synthago to try to enable these sort of technologies. So you've heard all day about industrialized CRISPR for accelerated discovery to clinic, and how Synthago positions ourselves as a company within the field. But what I really want to tell you about is kind of this full stack engineering approach that we can bring from a collaboration, pretty much all the way up of these different vertically integrated disciplines, from research to chemistry and bioinformatics, to make a genome engineering product that sits at a sort of intersection between our Halo platform and our research groups. So one thing that we've kind of seen throughout the day is that Cas9 is inherently uncontrollable. Some sgRNAs have high number of off-target sites, such as we've seen over and over again through technologies such as Singdarticide's GuideSeq. And we've also noticed that we have some potential to predict these high-frequency sites, but not always. We can't always rely on the bioinformatics to give us a clear view of the sites that we need to study in further detail. What we've also seen recently is that off-target events can lead to translocation events, and as you can see from this diagram on the left, there are very specific instances such as this MLL-AF9 translocation, which can actually cause leukemias within a mouse model. Really, this boils down to, once introduced, Cas9 RNPs are active until they're degraded by cellular processes, and how I like to analogize this to is sort of a runaway train. We put millions of separate Cas9 ribonucleoproteins into a single cell and then just wait for the cell to turn these over. And that extended amount of activity time can lead to the sort of off-target effects that we want to try to avoid in a clinical type setting. There have been various methods that have been used to try to control CRISPR. Some of the earliest ones split up the Cas9 molecule into the N-terminus and the C-terminus, and with the addition of rapamycin, these can form back together and enter the nucleus to create double-strand breaks. Some fantastic work by my fellow co-panelist Lucas Harrington also has described anti-CRISPR proteins, where it really shows the genetic diversity and the arms race between bacterial and CRISPR about how phages started to evolve and to mutate through these anti-CRISPR proteins to either do active site deactivation or also even completely dimerize with the Cas9 protein and inhibit DNA binding. Other groups have gone with more chemical synthesis moieties, where they've actually photocaged the sgRNAs, where they attach very specific molecules to the uracils within the protospacer of the sgRNA, and with the addition of light, these release, and now the Cas9 is actually able to bind to the target and make a double-strand break. We kind of took the opposite approach to this and said, hey, can we actually control the off rate of the Cas9 RNP system and make a sgRNA that fragments in response to light? So in this example, we have what we call a CRISPR off sgRNA that under normal condition actually behaves exactly as you would expect a normal sgRNA. Has full editing activity, you can prepare and you can handle it the same way. However, with the induction of light, this sgRNA scaffold would actually fall apart and render it completely inactive. Whereas this obviously would not happen with a standard sgRNA. I'll skip you some of the details about all of these sort of proof of concept experiments that we did and some of the further optimization, but what we found is that through the addition of two photocleavable moieties in the backbone at very specific positions, we created the system that indeed, with the addition of light, this sgRNA would fall apart, and you could actually, if you see at the graph on the bottom on the left, you could detect each of these individual chemical species. Now, the decision point to put two different chemical moieties in it was actually more of a safety switch. We found that by using only a single moiety in there, we were actually able to completely ablate activity, but the fragmentation was not nearly as consistent as we would like. And so by putting two in there, we could get a near complete fracture of the CRISPR sgRNA. Again, to make a little bit of a long story short, and you can look at all of this data in the Nature Communications paper that we published, but we identified that CRISPR-off is in fact able to modulate double-strand breaks within a population. And if you saw the sort of colors that we had on the previous slide, make sure to pay attention to those. So we have blue, which is our CRISPR-off sgRNA, which has not been exposed to light, green, which has been broken apart, and black and gray are our normal sgRNAs. But we saw that there is this huge delta in populations that had been either not exposed or exposed to light, meaning that we could get all or nothing. But we also hypothesized that we could potentially titrate the level of editing events within a population. And that's exactly what we showed in this sort of labor of love experiment, where we harvested each of these different populations 48 hours post-transfection, but actually exposed a different population to light every 2 hours post-transfection, and measured the level of editing activity. And indeed, what we saw and what's really cool about this is that CRISPR-Cas9 editing starts to more look like an enzymatic reaction where you have this very sort of exponential growth before you get saturated editing. And by adjusting the time post-transfection that you actually expose this population to light, you can titrate this level of editing to your desires. We also proved that these Crisproff sgRNAs are robust against targets. And so you can see, for example, with your ONPNS1 and your MIP targets, you're actually getting a very, very significant delta between completely on editing that's very similar to the level that we see with sgRNAs, and editing that's completely turned off 4 hours post-transfection. Now, some of you that are eagle-eyed might notice that some of these don't seem to work, such as the FAM163A and the FANC, but actually further experiments on the system showed that this is a really interesting site kinetically, where that the double-strand break and repair actually happens before 4 hours post-transfection, which is when we normally turn it off. So this sort of the kinetics of double-strand break formation and indel repair can happen very, very quickly. And we believe that you can use this system to start to really narrow in and start to focus in on what sort of kinetics are happening over time within an individual population of cells. We also made a very interesting observation that we believe that this system is able to modulate the on to off target editing ratio. And so what you can see is that on the top, there's the on target editing event, which happens very quickly. And then the edits sort of saturate at probably the number of cells that were actually successfully transfected. However, the off-target events actually have more of a lag period where it takes them a little bit longer to find those targets and to get going before eventually catching up and having a relatively more saturated editing percentage. We thought that maybe we could expose the CRISPR-off sgRNAs to light at these points to try to maximize the on-to-off targeting ratios. And indeed, that's exactly what we found, where with sgRNAs, didn't matter when we exposed them to light, they had the same sort of on-to-off target editing ratio that we would see at any times. However, if we exposed our CRISPR-off sgRNAs to light either 4, 8, or 16 hours post-transfection, we were still able to obtain very high levels of on-target editing while also mitigating some of the more prevalent off-target edits and completely ablating some of those that are not nearly as common. So far, we've told you about our CRISPR-off system, which is where we take an sgRNA and we're able to break it apart and absolutely shatter it. And how we like to think about this is a little bit like a light switch, where we have a molecule that's completely on, we walk into the room and we turn it off. However, could we also start in the opposite position, where we have a molecule that's completely off, we turn it on where and when we want to, and then turn it off all over again? And indeed, we've also managed to create this in a system that we're calling CRISPR-on. Not necessarily the best naming conventions. If you have a better one, please contact me, and we're more than happy to start that discussion. So how do we make this CRISPR-on light switch? And again, to try to, we're not going to go into all the details because it's only a 15-minute talk, but we found that by adding a backbone, with a backtrack, of RNA molecules across that perfectly matches the protospacer, along with the inclusion of 4 different light-sensitive molecules, we were actually able to completely block protospacer binding activity that when we shone light on this entire structure would fall apart and leave a fully functioning sgRNA. And you can see this very nice dose-response curve here at the bottom, where with increasing irradiation time, we have a decrease in the number of inactive molecules as detected by ESI, and we also have a corresponding increase in the number of active sgRNA molecules that are capable of making an edit. We again showed that these CRISPR-on sgRNAs are robust across targets, and so now here we have a CRISPR-on minus light as the green bars, and you can see that there's a much lower level of editing activity, if not completely ablated across a vast majority of these targets. However, when we turn them on, we're actually able to achieve levels of editing that are similar to a normal sgRNA that you would just transfect into your cell and use as typically done. But the pièce de résistance that we want with this is what we're calling CRISPR Lite, which is a fully tunable sgRNA. Now, the majority of experiments that I've talked to you before have this off-the-shelf available PC linker, which is based off of O-nitrobenzyl. But thanks to our really fantastic chemists here at Synthago, including Anastasia Kanita, we've actually de novo created a coumarin linker, which is based on a completely different photochemistry, but is still capable of linking. The PC linker is broken at 365 nanometers, whereas the coumarin linker is more broken at 420 nanometers. And what that means for us is that we have this really cool bioorthogonal shift where if you look at these optical densities and what kind of energy it's taken to break these sort of bonds, when we expose these cells to 420 nanometer light, we're only going to break the coumarin linker. However, this will not break the PC linker, and we're still going to be capable of using this. Now, combining these, we can create this CRISPR Lite sgRNA, which again is our fully tunable sgRNA. We integrate the coumarin linkers at the places where we want to be able to turn the sgRNA off, and we incorporate the O-nitrobenzyl-based linkers in the backbone where we actually want to be able to turn the CRISPR molecules off. As a proof of principle, we also wanted to show that we could enable orthogonal activation and deactivation of editing. So in this experiment, we transfected a CRISPR Lite sgRNA and just let them go. So if you see our normal sgRNAs, after 8 hours, we've accumulated a decent amount of edits. However, with our CRISPR Lite sgRNAs, we see 0 level of editing across at this time point. At this time point, we then turned on the molecule and let it accumulate edits for the next 24 hours. Indeed, what you can see is that when the molecule that we turn on has a very similar rate constant to what we're seeing with a normal sgRNA, suggesting that we're able to get the same level of editing that we'd be interested in with a normal sgRNA. More importantly, the inactive molecules are still completely inactive. We don't have any editing in this sample. At 24 hours post-transfection, we then turn the molecule off, and what you can see is we're actually getting a separation between the molecules that we left on and the molecules that we then turned off. And the levels of percentage editing does increase a little bit over this time due to indels that are in the cell that are currently still trying to be repaired. We've also shown this at multiple sites, such as EMX1 and STK3, showing that this is more of a generalizable solution for this fully tunable activation and deactivation of editing. So just in conclusion, there's a whole bunch of really other cool applications that we haven't quite told you about, such as this one on the right, where you're actually able to do, with just this simple upright microscope, as well as a thin film mask, you're actually able to do spatial patterning within populations of cells. And of course, we had to draw our logo on it. But really, CRISPR Lite, and what I hope you take away is that CRISPR Lite is a turnkey technology for anyone using Cas9. If you currently work with in vitro transcribed guides, if you currently work with synthetically modified guides, such as what we provide, you can immediately put this into your system and it'll be completely active. It enables a titratable levels of gene editing on a population scale, and that further optimizations allow users to maximize the on to off target editing ratios. We're also continuing to work on this, where future versions are getting longer and longer for both safety as well as penetrance into deeper tissues. We're also really excited about the potential to expand this to other nucleases such as Cas12As, Cas13s, or any of the other mini nucleases that you've heard throughout the rest of this session. So with that, I really just want to thank the team at Synthago. We have a fantastic research team here. You can see we've moved into our brand new building that we're very, very excited about and still building out the labs. And also open up an invitation that we're actively recruiting to work on these projects, to work on other cool, novel projects that are looking at pushing the boundaries of what we can do with CRISPR technologies. So if you're interested in these two, I do have QR codes for both scientists and research associates to work on this. Or if you're really just interested in Synthago, please visit us at www.synthago.com/careers. Thank you.